Initiation of macroautophagy starts when a cup-shaped membrane buds off the endoplasmic reticulum. This membrane requires a protein complex, including proteins ubiquitin-like kinase 1, short for ULK1, and other autophagy-related proteins to form a pre-autophagosome. It's worth mentioning that ULK1 is important for initiating the process of pre-autophagosomal formation and therefore initiating macroautophagy. AMP kinase is found in the cell and activates ULK1. A signaling pathway known as mTOR can become involved in this step. If no cellular stress was found, such as starvation, mTOR becomes activated. The mTOR complex acts upon the autophagic pathway by inhibiting AMPK, and therefore inhibiting ULK1, which in turn inhibits the autophagic pathway. The second step of autophagy is nucleation. ULK1 from the previous step activates Beclin-1 through phosphorylation. This complex acts as a class 3 phosphoinositide 3 kinase and phosphorylates PIP3, known as phosphatidylinositol bisphosphate, from PIP2. The increasing concentration of PIP3 leads to the recruitment of proteins to the pre-autophagosomal membrane. These proteins recruit other proteins with them as well, such as P62 and NBR1. These proteins act as partial selective receptors for different substrates in the cell. The third step is elongation. It starts by cleaving a protein called pro-light chain 3 into light chain 3-1 by cysteine protease ATG4. Then, light chain 3-1 becomes conjugated to PE to form light chain 3-2. During this conversion of light chain 3-1 to light chain 3-2, the autophagosome continues to elongate and form a complete and mature autophagosomal vesicle. It continues elongation until the contents are completely sequestrated within a fully formed double membrane autophagosome. So far, we mentioned 3 steps: initiation, nucleation, and elongation. So now we move on to the 4th and 5th steps, which are fusion and maturation. When the autophagosome matures, it can fuse with the lysosome through snare proteins. Once it fuses with the lysosome, it forms what we know as an autolysosome. After the fusion, the autophagosome releases its content into the lysosome, and these substrates are then degraded by proteases. And then the recycling begins, and this is the whole point of autophagy. Thank you for watching the video. Please like and subscribe, and don't forget to support us in the next videos. Thank you.